CCNTCSAGBHYK question mark.
Bingo.
I feel exactly like Blast is jamming us right now. Basically, first we want to get a sentence, right? We want to get a sentence of at least 30 plus letters. That means that whoever rolls the first one, it's a capital letter. It's the beginning of the sentence. Sentence that says you. Whatever she rolls is a capital, right?
I want that. GAT.
So we have a capital G. This is like a game show, right? We're going to make a genetic sequence.
Okay.
One letter at a time. Do you want it to be GAT or GAC?
GAC.
GAC. Okay. There's so few Q's. I mean, I think they chose Q for glutamine because it looks like a G, but it's not glycine, which already has the G. So I said you can substitute O, but for the database we need to use Q, so this could be GGQ or GGO, and you have to roll one.
Yes, go for this.
AAA.
And then we should probably get used to saying these. What is that? That's lysine, right? Comes out of yeast that's made genetically to create more healthy lysine than usual. Oh, I like
Q or O?
Yeah, Q or O.
every time you find the sort of rule base, if you look closer, there are eccentricities. It's just built into nature. I like life. So, okay, who's next? We got a lower Bill the strat. Pick up.
Oh God, we played there. C. Let's go THC.
Cysteine.
Cysteine?
Is it? Okay, give me a drum roll. This is high risk, high stakes. Oh, we got another cysteine. TGC. Wow.
AAT.
AAT. Is this like a mirror phase that we're in? Where we're like emulating our own transcription of our own genes? Or is this like a power trip? Or actually, is this like a drag queen? You know what I mean? Are we putting on nature's clothes? Or is it starting to be? Oh,
C.
we're getting a lot of C's.
TGC.
TGC.
TCC.
TCC.
It's an A.
Hey!
GCG.
GCG. A G. Whoa. V. V. It could be a U. And then what valine do you want? What triplet?
GTA.
I don't know. We'll do a few more and then we'll type it in, right? Okay. So really, we should keep going until we get a stop codon. Shall we, for the sake of convenience, decide which stop codon? Do we want a period, an exclamation point, or a question mark?
Question mark.
So TAG, question mark. Our sentence is capital D, D, Q or O, K, Q or O, Q or OA, CCNTCSAGBHYK question mark. So the first assignment for the continuation of this picture is right next to that, is to draw what that says as an image.
Okay, so now we have to make words with this, right? Yes. Make sense of this.
I started with a D-like figure, and from there it just evolved.
I had to think of the word dock as a shipping dock. in the beginning we have the DDOK, and I thought the ship is a container ship, and it's like holding all the DNA genes and strings, something like that.
Het moet ook een beetje licht kalend zijn met
The
een
dodo with the kooky cunt
brilletje.
being figured in the interface of
Die haai moet dan hier ergens staan, hè?
Shop CS and the sausage.
Een lange sloot papier met allerlei allemaal cijfertjes. Allemaal cijfertjes. Ja.
Not organs, like orifices.
Mucus producing bacteria living on an orifice. Oh, okay. And does it relate to the text for you?
Yeah, it does. Yeah.
You are a data miner. That's cool. So now what are you drawing? This is the
So
accountant?
this is the accountant in here. He's in his dark room.
Yeah.
Just doing some calculations on the way, developing some pictures.
You can draw a picture of anything.
Yeah, I was just.
Yeah, the void, it works too. You're allowed. Yeah. There's no message. It's possible.
Yeah.
QKQ is
Yeah.
quack.
Quack, quack, going very fast.
Okay. Maybe those are those redundancies. They're a little different, each one.
Yeah, but from here I got the idea of chocolate, cacao, cocoa.
And this is a ship full of DNA, and it's being closed into the station. It can send signals or something. I don't know exactly.
More interesting at this point is something called sequencing. The way scientists used to figure out a DNA sequence was by labeling A, T, G, and C with radioactive isotopes. And the radioactive isotopes would separate out the base pairs, one base pair at a time, and put them on X-ray. You'd get an X-ray, and it would look like this. It looks a lot like DNA fingerprinting right now, right? Much like what we're doing here in this lab right now, I have been in labs where they're trying to isolate a gene. How do you think 10 scientists that are on the same paper agree what this is as a sequence? You know what they do? They sit down together in a room and they go through this, just like what we're doing, all 15,000, and then they put them together next to each other and say, is it the same sequence? Because if all 10 of us got the same sequence, then we can publish, right? This is the kind of data they get, right, on a computer screen. And it's very much like what we're doing, right? In other words, if we zoom in, you can see underneath it, right? See down there? Last is the basic local alignment search tool. It's an online software. It's free for people to use. It's a part of the community of mind, right? These are what are called workhorses. The workhorses of molecular biology are the organisms that genomes got sequenced first, that they studied in the lab, and that are mirrors of us. In other words, they use them to try to understand humans. Of course, these are the totem animals of molecular biology, right? They're called workhorses, and they're heavily studied. If you just type in BLAST and NCBI, BLAST is the basic local alignment search tool, which finds regions of local similarity between sequences. Program compares nucleic or protein sequences to sequence databases and calculates the statistical significance of matches. It can be used to infer functional and evolutionary relationships between sequences, as well as help identify members of gene family. This is what we want to try. I'm trying a little bit of a different one. PsyBlast. I chose this for you, Gina. It's called PsyBlast. And I'm not sure if it'll work. I'm not sure if it works, but we'll give it a whirl, okay? Oh, we press here, right? Okay, hey, we got some. Oh, it says no hits found. All right, let's see if this works. Oh, it says that's not protein, that's DNA. So we got it right the first time. So I'm going to do this like, I'm filtering, maybe we should filter true instead of false. If you get a result and there's nothing to compare to it, that's good. In other words, we found a sequence that isn't in the database yet. Yeah, no one's seen this before. It hasn't been found. All the genomes that they sequenced, and we saw there's like 30 or 40 some other major workhorses, right? So this actually is important. This is a company. You're a partner in gene synthesis. I contacted them and I said, can we get gene synthesis and maybe even get what we have here written into a plasmid so we can put it inside of a E. coli and have the E. coli reproduce it. And they said, send us the sequence and we'll check it in our database to see if it's pathogenic. It may not coincide with any known pathogen, but it also doesn't coincide with any known sequence. So how can we risk assess since it's based on chance? We'll check it out. Anyway, we're going to send it to them, just so you know. Hi, we have an unknown sequence. And give us an art student price to manufacture the plasmid with this sequence in it. What is a plasmid? Anybody know? A plasmid is a circular piece of DNA that E. coli use to email each other with. They email genes to each other through plasmids. It's infectious. Oh my God, it's the weirdest thing. Most E. coli in the lab is F negative. F positive means that the E. coli has hair. There's hairy E. coli and E. coli that's bald. And the hairy E. coli has hair called F. pili, which is why they're F positive. And the F. pili are hairs, but their hairs are hollow. And when a hairy E. coli reaches one of its F. pili hairs out to a hairless E. coli, it shoots a plasmid down the hollow hair into the other E. coli, and that E. coli starts to get hairy. Yeah. So it's a kind of sex. If you could imagine, every time a man made love to a woman, the woman would turn into a man.
Homo Loki.
Homo Loki, yeah. The Homo Loki. Okay, here we go. We're sending, right? We're sending them our sequence. I'm going to take down this one. You really have an unusual sequence, huh? Everybody take off this. I don't trust this because it's not working at all. Should we try the cross-species megablast? Wow. Something's wrong. I feel like we're being jammed by blast. How is this possible? I mean, come on. I do think we might be being jammed by blast. Let's view the report. I'm going to try once more. I'm going to blast home. We're going to do a protein blast. The protein data bank. We want
Use default settings everywhere.
And it's pulling up the transcript. The reason it takes a while is because the transcript, it shows where it is in the human chromosome, and you can click into the chromosome for different genes. So we're looking for PAN2, right? This one was transcribed by Celera, a company, right? A company that actually challenged the US government public genome sequencing project to a privately run corporate. Ended up working with them, but only reluctantly. And he still charges. So eventually, if you get too deep in this, you're going to have to pay Craig Venter. Please draw Homo sapiens pan-2 poly-A specific ribonuclease subunit. How do you date them on art? Yeah? Sure. Is it without algorithmically? Yeah. Okay, so we actually have rolled a sequence that has has some homology with BP 446853 full-length enriched swine cDNA library for the adult liver. Suscrapha cDNA clone LDR 010031G085 negative mRNA sequence. So, I mean, you can actually like research this. Suscrapha, you can look up, but also it happens to have a PubMed number and a contact person, right? Who happens to be a Japanese scientist in the Animal Genome Laboratory, Genome Research Department, in AgroBio, right? So you can actually get this guy's email or phone number. Here it is, right? There's his email. You can email him and say like, hey man, we rolled a sequence today and we got a homology for your, well, we don't know exactly what this is, right? We drew pictures of it, but maybe you could tell me about it. This is actually funky research, right? Like a strange way to research. Because this guy's waiting for someone to say, I'm interested in your research. He wrote that paper, he published, he will not perish, but he's like completely ignored. There's only 12 people in the world that understand him. And you write him and say like, I drew a picture of your full-length enriched swine cDNA library of the adult liver. Do you want to see it? You know, do you want to see it? He's going to be like, yeah. So let's draw that. I'm going to try to draw a full-length enriched swine cDNA library of an adult liver. Take a couple of minutes, all right? It can be quick and dirty.
Black.
Well, take a look at this. They've got the original sequence there. Doesn't look that different from what we've been doing, does it? You notice? If I pull this out and go into Microsoft Word, I may find our sequence. You understand what I'm saying? It's a strange world. How about that sweet orange leaf? What else have we got?
Koffie Arabica.
Japanese lotus pod, right? Tilapia.
Fish.
Okay, so let's draw some tilapia liver. So look, we've got lotus, coffee, absinthe. All of a sudden we're making waves. And tilapia. This sounds like a good dinner. You know? I have a contact in Japan. I guess it's just the pod, right? And they made a clone of it. So we have it in the human, right? And we have it in
The coffee. That's why humans drink coffee, of course. That's
To fill up their cells.
So, let me see, I want to do this last one more time with the full sequence because I feel like I'm cheating, even though there's no such thing as cheating because this is an art class.
GAC, CAA, AAA, CAA, CAG, GCG,
Wait, hold on,
TGT,
CAG, GCT.
TAT, TAT, AAA, T,
Okay. But just because your sequence coincides with homologies, etc., doesn't mean, even if you find out actually one of the pathways of the expression of this gene into a protein and what it doesn't mean that you understand what's going on. Scientists would be out of a job if understanding came full force, right? In other words, like, we've got to keep checking. Our sequence is all the same. It's a cDNA clone of Lattes calcarifer. I'm going to zero in on it, but I'm also going to look it up. Barramundi perch actually seems to be our sequence's familiar, right? For real. A barramundi perch. Lattes calafere. In this guy's lab, we're actually going to have to contact these people. In Malaysia, right? This guy Adura is our new friend. Like, he's going to hear from us. You guys know that, right? After class, I'm going to write him. I can write him right now if you want. You guys want to help me compose a letter? Yeah. Let's write him a letter. This is our animal familiar of the day. We rolled it right here in class. This is the barramundi perch. And this is the barramundi fillet. And this is the giant barramundi. So this is actually the fish that has the sequence that we rolled here. That's on our paper that we translated to like dodo, cookie, cunt, adobe, CS, sausage, fly. Take some time to draw not just the barramundi perch, but we're gonna have to find out where in the barramundi perch this sequence, the cDNA library or this messenger RNA has been found to be used. I'm not sure if it says. We gotta inform ourselves. Okay, so this is the paper that it's written up in. Is that possible? It's from Gene 411. It's a brain transcriptome. It says, identification and analysis of a pre-pro chicken gonadotropin releasing hormone 2, otherwise known as pre-proc GnRH2, precursor in the Asian sea bass, Lattes calcarifer, otherwise known as the barramundi, based on an EST assessment of its brain transcriptome. That's some heavy stuff. Do you think we can draw that just for 5 minutes? I know we've been doing a lot of drawing. This is this is gonna finish all of our drawings. Pre-pro chicken gonadotropin releasing hormone precursor in the Asian sea bass. I'll pull up the fish on the side. Gonadotropin. It's a brain hormone, I believe, but let's check. I'm gonna just add to your drawing. This is I wanna let you know this is called alternative research, like research methods for weirdos, but gonadotropin, half of what I know about biology comes from random walking like this. Luteinizing hormone, so yeah, it's actually decides your gender to some extent, right? It's secreted in the brain, in humans, in the pituitary gland, right? So if you're studying gonadotropin in a fish or in a chicken, you're actually studying it maybe to get some extra for people with hormone deficiency. They do. I happen to know, like, it helps with the formation of the genitals, but also there's a third human gonadotropin produced by the placenta during pregnancy. It's taken from the urine of postmenopausal women. Well, it says it can stimulate spermatogenesis, the making of sperm. Yeah, it can make sperm and eggs for ovulation and help with fertility. Strange. But anyway, so throw a little gonadotropin in there and maybe some sperm genesis and ovulation, right? Coming from this chicken fish EST. And it's in the pituitary gland in humans, so is it in a homologue for the pituitary gland in the fish, right? An ancient evolutionary pituitary gland that has been steadily developing in both fish and humans from perhaps it was a separate organism before the brain formed, right? Glands in our brain, what are they doing there? Where did they come from?
It may be.
What should we say? The sequence that we rolled happens to be in the sea bass in Malaysia, and it says using a novel library of express sequence tags, or ESTs, from the brain tissue of the Asian sea bass, we characterize the brain transcriptome for this, get this, quote unquote, economically important species. Forget the brains, but obviously we have found a hormone-related brain neurotransmitter, a gene that relates to hormone release in the brain that also relates to fertility.
Well, we have
DD. So we got a double dog attached at the behind with some quacking ducks, and they're inverse, so they're one on top of the other. We have lots of accents of different languages spoken by the quacks and the dogs. Here we have the Homo sapien pan squared with a ribonucleus subunit, lots of subunits and subunits. And this is just some kind of brain mapping texture with a transcriptor fish swimming out of it. And then some coffee stains.
Yeah, I translated all the letters in colors and forms, and they started growing out, randomly. And sometimes the purple D's came out of a green line, but then they also come out of a red line. And the orange D's came first out of red lines, but then started growing out of purple D's. It's all a new kind of chocolate cocoa bean. But then when you split it, the inside is different. So maybe we get a new taste if we make chocolate out of it. I thought there was a kind of chicken in the fish, which it poured out hormones. I think it's in the stomach.
My sentence is a container ship full of crates that hold different kinds of DNA. This station is trying to give meaning to what's all on the ship. And then the fish, it's like loose from it, but don't have, this is more abstract, I don't know. Cannot explain this.
He also saw the dark room of the accountant. That's basically what the sentence says here in Dutch. The swine liver here. We have the, or is it like the messenger RNA or the ribonucleic acid, something like that. And we have the fish here. So yeah, that's basically it. So we kind of figured out what he was doing later on, because he was obviously developing all those pictures.
Oh, definitely. That's what the accountant was doing in his work.
Yeah.
This is the pan gene. That's pan for me, is like the satyr. So that's pan 2, that's the little pan that lives in your head. But this is the gene, this is the gland in the fish's brain that produces the luteinizing hormone that adds fertility, as you can see, maybe through pan 2, comparable to the chicken, similar chicken hormone producing. That actually this hormone produces sperm and eggs.
That is that he divided the painting in very strict regions of difference. One with the colors, one with the dodo, one with something that has to do with sequence. And it all comes together referring to the messenger DNA, messenger RNA, which is traveling from one strand to the other, compiling the protein.
From this, I tried to remember what I saw here and how to draw the sentence in this circle. Yeah, I saw them like star signs.
Yeah, they are constellations, right?
Constellations, yeah. And as a matter of fact, this way of reading is very ancient. Like if we see them as star constellations. So I was looking at this like, what is the perspective if we draw it down like this? Is it from the human perspective that we see the code of the DNA, or can we see the code from within the DNA? So, yeah.
I like
I was just trying to take the perspective of the specific fish brain.
And the three-dimensional shape is only when it's crystallized. Actually, these proteins move all the time.
Three-dimensional.
They're three-dimensional and they're in motion. Yeah.
I liked the idea of that we had the fish because it's an underwater, and so in the water you don't maybe know that above the water there is another world.
You clearly see its hormone being released inside of the brains of our perch. This one was just a free association, but you clearly see subunit 1, which we were supposed to draw. This was like the swine thing, so it's very amorph, except for its nose. And I thought, Q, that reminds me of cocorico, right? Of a cock. And then I read further, and then it suddenly said UHY. So I said, you ate why you eat a cock.
We don't know exactly what we're doing when we alter genes, okay? I do know that if you put your finger in a river, you change the river. If you decide not to put your finger in the river, you're also doing something that causes change. By refusing to act is an action. So it's actually a very difficult situation that we're in. There is no way for us to not affect the world. But it's enough, I think, to teach right now at least, that chance is built into life. So when you mess with chance, you mess with the muses, right? That's okay. There's a long history of heroes and victims messing with the muses, right? But be aware that you may not know what you're doing, right? I guess the meaning of these labs, for the most part, is to both demystify and remystify DNA at the same time, but also to let you know the complexities. So when someone says, well, there's a gene for schizophrenia and we should just get rid of that, that you say like, well, now hold on. You know, genes aren't just one-to-one. You know, just getting rid of that isn't a simple procedure, and you're affecting, the entire human genome. And without schizophrenia, we wouldn't have disco, or like really cool free associations, or art for that matter, right? So slow down, that kind of thing. So I hope I can transmit that to you.
This is the satay kambing made of the goat that was used for the tissue culture lab last Wednesday. Well, it was Tuesday, sorry. So it's fermented a little. Only organic stuff. Whoa.
Want a beer?
Yeah, second. Oh, that's alright. Put it there. I can put some in the satay.